It's my pleasure to introduce our research guest lecture today. So you're here today from Dr. Susan Gray. So Susan's an assistant professor at OSU School of Environment and Natural Resources. Been here about, what, a year and a half?
9 months.
Yeah, excellent. Well, in the short 9 months that Susan's been here, we've already we started collaborating with her on some wetland projects that are happening along the North Shore of Ohio, looking at some recovery of wetland fish species to new hydrological changes in those wetlands. So she's been active in that. But graduated 2007 from your PhD in behavioral ecology from Simon Fraser University, which is in Vancouver. And then post that, you had 2 postdocs running simultaneously. So one was through McGill University in Montreal, and the other one you had from the Department of Fisheries and Oceans Canada. And so both of those postdocs working on similar stuff to what she's going to talk about today, but turbidity, so sediment loading, and it could be algal turbidity too. So turbidity in ecosystems and how that impacts fishes. So some cichlids from Africa and also some shiner species from the Great Lakes. But her talk today, the effects of turbidity on imperiled Great Lakes fishes, and as we always ask for both our guests in research lecture, she's going to give you a little glimpse into her world. So what kind of decisions in past she took to kind of get to where she is today. All right, everybody, welcome to Susan Gray.
Okay, well, while that's getting set up, I'll just tell you a bit about myself. I grew up in Nova Scotia, Canada, on the seashore, playing with little fish and shrimps and whatever I could get my hands on in the ocean. So my sort of love of biology and water started really early. My parents always said I learned to swim before I could walk. I'm more comfortable in the water than I am on land. And then my path to being an assistant professor at the School of Environment and Natural Resources at OSU was fairly direct in terms of knowing I wanted to do research from a really early age, and I wanted to be in water. And at one point, I went to Australia on a field course during my undergraduate time, between 3rd and 4th year, and I found out about the world of tropical biology, and that was it. I was going to be a tropical biologist forever. And I followed that path, and I was going to do field research, and I just found positions where I got to do field research. My master's was actually in Northern Ontario, not particularly tropical. It snowed one time in August, and I thought, this isn't good. So for my PhD, I chose University as the place where I would do my research, and I just kept following that path. Chose a postdoc that allowed me to work in Africa, where I'm still working today with collaborators. But during that time, I was also approached by a manager, Department of Fisheries and Oceans Canada, to work on a more local project, endangered species. And I thought, oh, that is so cool. So I did that. And I think the point I'm trying to get across here, especially to the undergrads in the room, is look at those opportunities, think about what you're passionate about, and just follow it. And I know that's how I got where I am today. So I'm going to talk to you about some of this work actually I did as a postdoc, looking at the effects of turbidity, or suspended particles in the water column, on imperiled Great Lakes fishes. And I'm going to take just a step back and thinking about fresh waters in general. And I might be preaching to the choir a bit, given some of the REU work I just heard about. You guys know a lot more about algal blooms and such things than I do. But freshwaters are among the most seriously threatened ecosystems on the planet. Humans, have disproportionately settled near freshwaters, modified riparian zones, contained, diverted, polluted freshwaters, and extracted freshwater resources. What we have to remember when we think about our freshwater and our freshwater resources is what a tiny fraction of the Earth this really represents. So this sphere, the one next to the Earth, represents the volume of all the water on Earth. That includes oceans, atmospheric water, ice caps. The smaller sphere is all liquid fresh water, but that includes groundwater. So when we think about the water found in lakes, swamps, and rivers, it's this little tiny bubble. And if you pop that bubble, let it disperse over Earth, it's going to cover less than 0.8% of the Earth's surface. Okay, so that's the tiny fraction of the biosphere covered by available water for lakes, rivers, and swamps. But in that tiny portion of the biosphere are over 10,000 species of freshwater fishes, almost a quarter of vertebrate diversity. That's a lot of diversity in such a small space. Given the human alterations of freshwater ecosystems, it's really no wonder that about 30% of fishes are threatened due to human activities. So fish face this challenge of being restricted to this small portion of the biosphere, as well as the human impacts that we're having on them. For example, hypoxic or low dissolved oxygen events that lead to massive fish kills, over-harvesting, degradation of habitats, and pollution. So they're facing space constraints, all of these environmental stressors, and they also have that physiological constraint of not being able to just get up and walk to a cleaner catchment, right? They're constrained to life and water. My research has focused on understanding how do fish respond to such severe and rapid alterations to their environment. So, big question, broad, how do fish respond to environmental change? They can do different things. Some species can cope with severe and rapid environmental change by adapting genetically or plastically to the stressors, or they can move away from the source of the stressor if there's some clean water elsewhere. But at the other end of the spectrum are those fishes that either can't respond to the stressors or respond too slowly. They can't cope with environmental change, and in that case, we might lose fish diversity. In order to sort of address this broader issue, I decided initially, and this was during those postdoc years, to start focusing on a single environmental stressor, increased turbidity, so increased sediment or algae in the water column. I looked, and I'm continuing to look at a species of African cichlid that is found throughout the Nile River Basin of East Africa, and it actually seems to be coping with increased turbidity. So turbidity caused by human activities like deforestation and pollution. We see population-level divergence and some really interesting adaptations to turbidity. I've also been looking at this other end of the spectrum of fishes that don't seem to be coping with increased turbidity. So I'm looking at imperiled fishes in the Great Lakes. Imperiled meaning we're losing populations and species in many cases. And that's what I'm going to focus on today, is sort of this end of the spectrum and the fishes in the Great Lakes, since we're sitting here on the shores of Lake Erie. I'll focus on that work, and I can talk about cichlids later if anyone wants to hear about that. So turbidity. There can be natural turbidity that a lot of fish species and populations have evolved in naturally turbid waters. But turbidity is increasing globally due to human activities. As I mentioned, deforestation, shoreline development, leading to sediment runoff. Dredging causes a lot of suspended sediment in the water column. We can also have organic turbidity. Lots of talk around here about algal blooms. That's a form of turbidity, suspended algae in the water column. What is all this turbidity going to do as an aquatic? Oh, I forgot about this picture. This was released by NOAA, the 2013 algae bloom in the western basin of Lake Erie. So it's algae here, but there's also sedimentary turbidity that can be an issue in this area of the lake as well. I'm sure you're all pretty familiar with this. So what are the effects of turbidity on aquatic ecosystems? I have a very fish-centric view of the world. So this talk is going to be pretty fish-centric with respect to what the effects are of turbidity. So there can be indirect effects for fish. The shading of benthos, blanketing of the benthos if the sediments fall down out of the water column, that can affect reproduction in fish if they're laying demersal eggs, that sort of thing. It can remove macrophytes so that there's less shelter, less places to avoid predation. So those sort of indirect effects of turbidity. There can also be very direct effects of turbidity for fishes. So reduced visibility as light is scattered and absorbed by the particles in the water column. Vision can be reduced, and that can have big implications for finding food, finding mates, avoiding predators. Another direct effect can be the clogging of gills and the abrasion of sensitive gill structures. what that can do is make it really hard for the fish to uptake oxygen. So it basically can put them in a state of hypoxia if they can't get that oxygen through their gills. Okay, so some of the direct effects. And we're going to go through these in just a little bit more detail. We're going to talk physics for a minute, so everyone just relax. It's okay. It's colorful. I wanted to talk about light underwater. Okay, so in a clear lake or ocean, you can have light going down below 75 meters. Okay, we do notice that there is attenuation of different colors of light. So long wavelengths, or red and yellows, are going to be lost very quickly due to scattering and absorption at the shallower depths. And as you go down, what you get is sort of the blues and greens at depth. In fresh water with green algae, you can see that light is lost at a much shallower depth than in clear waters. And you've noticed that the short wavelength lights get scattered, they get lost at shallower depths. And what you have at 25 meters, for example, are the greens and yellows. In fresh water that is turbid due to certain types of sediments, this is just an example, 3 meters, no more light. And the light that's left is red shifted. You lose all those short wavelength light. So basically, we're going to have changes to the visual environment. What does that mean for a fish? I know a lot of you think about, when you're thinking about algae and turbidity, you might be thinking about the limnology and that sort of thing. I think about the fish and what they're seeing and what they're going to do about it. Let's talk about signals for a minute. Here's a fish. We're going to call him a male, nice bright yellow male. If this is his color signal, that signal, the efficacy of that signal is going to depend on the visual environment that that signal is sent. It's also going to depend on the receiver of the signal. So in this example, let's say we've got a female, she's looking at this male. What are her eyes sensitive to? What colors can she see? Does she see this male as conspicuous? He's yellow, he stands out against that blue background. So she might say, hey, I want to mate with you. You're bright, you're conspicuous. So that's great. Oh, dear. Something went wrong. Wow. He was so excited about this female in this situation, he exploded. So this is a competitor fish. He might see this signal, pretend that signal is as conspicuous. So now this guy has some competition. So we've got some within-species interactions happening, all associated with this visual environment. Now, I really hope this one's not exploded. Oh, okay. Oh, the predator is good. So the predator might also see that signal as conspicuous, depending on its visual capabilities. And so now we have a situation where we have within and between species interactions, finding mates, reproduction, survival, looking at predation. So the visual environment's really important. Let's say we go outside and we dredge the channel in front of Gibraltar Island, and we stir up a whole bunch of sediments, and we get turbidity. That signal may no longer be conspicuous. It could be masked by the noise caused by the scattering of the light. Okay, so that's going to influence all of these interactions, the within and between species interactions. What is that going to do to the populations of fish if they can't find the right mate, if they can't avoid a predator? These are the sorts of things I think about a lot in all of the systems that I work in. Now, I changed this up a little for everybody here. So I added the human dimension. Okay, so we have our visual environment. We've got a fisherman. He's out there with his worm on a hook, and we've got the predator that he's trying to catch. We throw in that algal turbidity that everybody's talking about. So this is a different color shift in that visual environment. What happens to this system under these conditions? I don't know that I'm going to tell you the answer to that, but it's an interesting question. Okay, so I also talked a little bit about a direct effect of turbidity on clogging gills, abrading gills, and how this might affect the ability to take up oxygen, aerobic capacity, and then the ability of the fish to perform energetically expensive activities like swimming, avoiding predation. This is just a quick study I wanted to show you as an example. This was Sutherland and Meyer in 2007. They exposed spotfin chub juveniles to different levels of sedimentary turbidity for 3 weeks. What you see here is the control. Normal gill structures, sort of delicate gill lamellae. And this is what happened at the highest level of turbidity, 500 milligrams per liter of a clay suspended in the water. The gill lamellae are thick, they're close together, they're damaged. The ability then to take oxygen is going to be compromised. Growth is expected to be reduced as is condition. What that means for then a population of fish exposed to this kind of turbidity is not necessarily known, but it could be very detrimental to the population. Okay, so I'm going to now tell you about some of this work that I've been doing, and again, why I started doing it. Because I was working on cichlids, I was going off to Africa and understanding how they were dealing with sedimentary turbidity. I was approached by Dr. Nicholas Mandrake at the Department of Fisheries and Oceans Canada, who works with aquatic species at risk. He said, hey, you do this stuff with cichlids and turbidity. What do you think about Great Lakes fishes? Like, well, I like Great Lakes fishes. And he suggested that the problem that they're having is that they know that turbidity is implicated in the decline of many fish populations in the Great Lakes, but the data on turbidity thresholds was lacking. And as a manager, the government was saying to him, well, you can't tell us how much turbidity is good or bad for these fish, so we won't bother helping these fish. I'm paraphrasing a little, but that was the basic interaction that was going on. So we took the approach of looking at sublethal effects, direct effects of turbidity on some imperiled fishes, comparing species at risk with non-threatened relatives to kind of try to get an idea of how fish were responding to increased turbidity, and also looking across life history stages within species. So the first project that I'm going to tell you about was looking at imperiled Neotropis shiners. There are many Neotropis shiners in Ohio. The ones I'm going to tell you about, as you'll see in a minute, are actually extirpated from Ohio, but we still have a few populations left in Canada. And this was postdoc work at McGill University with Loren Chapman and with Department of Fisheries and Oceans and Dr. Nicholas Mandrake. So the fish that I'm going to tell you about, shiners, some of you might know a bit about shiners. These particular species tend to be schooling or shoaling species. They like really clear, heavily vegetated waters. And in those waters, then, we expect them to behave normally. So they're in their school, they're swimming at their normal speed. They're doing what we expect them to do. What do we expect them to do under turbid conditions? We have some evidence from other fishes that under turbid conditions, schools tend to break up. They become looser. Schooling in fish is really important for avoiding predation and finding food. Okay, so it can be detrimental to those species to have looser schools or no schools. Okay, so I've just, as an example, now this fish, he's all by himself. Maybe his gills are abraded. He can't take up enough oxygen. So his swimming performance is decreased. He's a bit slower. That leaves him vulnerable to mortality through lowered condition or predation. My predator got more advanced. He didn't talk. So these are the fish that I was working with: the Pugnose shiner, bridal shiner, black chin, black nose, and the mimic shiner. And I've depicted them here along a reported turbidity tolerance gradient. So these turbidity tolerances are basically based on presence-absence data of large surveys of where the fish are found and are not. The Pugnose shiner was listed as endangered in Canada in 2003. There are 4 remaining known populations, mostly in Lake Ontario and the St. Lawrence River, so sort of up in this area. They are extirpated from Ohio as of, I believe, 1930-31. Bridal shiner listed as of special concern in Canada. They have never actually historically been in Ohio, as far as I know. Their sort of western range is over here. Black chin shiner, black nose shiner. are actually fairly common in this area of Lake Ontario and the St. Lawrence River. We find them not everywhere, but in clearish waters. Mimic shiner is reported as being somewhat intolerant to turbidity, but where we were catching them in the St. Lawrence River, turbidities were fairly high, at least relative to where we find the other species. So, Pugnose shiner, extirpated from Ohio. Bridal shiner was never around. Black chin shiner, black nose shiner, both extirpated from Ohio. The last known records were in the 1980s. Mimic shiner, from what I understand, used to be very common, but populations are reportedly declining in Ohio. So I'm going to talk to you about a bunch of fish that aren't in Ohio anymore, except maybe the mimic shiner. So we asked two fairly simple questions. Does turbidity alter schooling behavior in each of these species, and does turbidity decrease swimming performance? So for the first study that we did, we collected all 5 species from populations along the stretch of the St. Lawrence River near Kingston, Ontario, where the lake drains. So we collected adults, we brought them back to the lab. Species were kept separately, and we had 10 to 15 fish per tank. There were 3 turbid treatment tanks and 2 clear treatment tanks due to logistics. And basically what we did is in the turbid treatment tanks, over a 2-week period, we slowly increased turbidity. And during that time, we observed both types of treatment tank, controls and turbid, and observed schooling behavior. We wanted to see if there was a change in schooling behavior over time amongst the different treatments and then amongst the species. Now, I'm going to mention that the highest turbidity that we reached in these experiments at the end of about 2 weeks was 12 NTU. So some of you who know, yeah, I know the laughing, and I can just see the reviewers' comments now. That's really low turbidity. You can still see through it. There's not a huge change in the visual environment at 12 NTU. But I'm going to show you that there were behavioral responses to that low level of turbidity. I will also note that the 4 populations of Pugnose shiner that we know about that still exist in Canada, average turbidity over several years was always less than 2 NTU. Okay, so where we find them in nature still, it's clear water. This test was still fairly clear water, but higher than in nature. So I'm going to walk you through these graphs. I'm just going to show you 2 of the species as an example. So the Pugnose shiner, which is endangered, it's been extirpated from Ohio for quite a while. What we have on the x-axis is trial day, and then we have the mean proportion of fish in a school on the y-axis. Blue markers and lines are the clear treatment fish, and in brown are the turbid treatments. So if we just look at the Pugnose shiner to start with, over time, in the clear treatments, there was an actual increase in the amount of fish in a school. The proportion of fish schooling in a tight little school in the tank, it increased over time. And what we think this is an acclimation to the experimental observations. We had to go in to the little chamber every day and sit down and sit for 30 minutes and then start our observations and watch the fish. So we think that this increase in schooling behavior might have been some response to the experimental conditions. What's interesting, though, is that the fish that were experiencing increasing turbidity during this same time, exact same procedure, showed no response, and then at the later stages actually decreased their schooling behavior. So there were fewer fish in a school. They were spread out around the tank and just sort of moving rather erratically around the tank. So a divergence in schooling behavior between the 2 treatments. The black chin shiner, I've used it as a representative because it matches what the black nose shiner and the mimic shiner did. Essentially, there wasn't really any difference in schooling behavior between the 2 treatments, at least at this really low level of turbidity. We didn't go beyond that. We were having trouble seeing the fish. So that's where we stopped the experiment. We did want to compare between the species. So what we did was took the difference in the proportion of fish in a school on the very last day of the treatment. We took the difference between these last 2 points for each of the species, and we plotted that along our reported turbidity tolerance. And what we found were these significant differences in schooling behavior between clear and turbid treatment, in the endangered Pugnose shiner and the special concern bridal shiner, but not the other three species. So does turbidity alter schooling behavior? Something's going on in the Pugnose shiner and the bridal shiner. With increased turbidity, the fish just didn't respond the same way the fish in the clear water treatments responded. Is it a visual thing? Is the visual environment changing, that they're loosening their schooling behavior? We don't know the answer to that. It leads to a lot more questions. What is going on with respect to schooling behavior? It's a clear result, not necessarily able to explain it with the experiment that we did. Lots of room for future work. So the next question, does turbidity decrease swimming performance? So the idea from this question, if you remember, comes from the idea that turbidity might affect gills, respiration, uptaking oxygen, and aerobic capacity. So what we did was we held the fish at their final experimental conditions after the behavioral tests. So the clear treatment were kept in clear water, and the fish were kept at the highest, about 10 to 12 NTU, for about 2 or 3 months. We gave them a lot of time to acclimate to these different conditions. We then tested for critical swimming speed in this standard swimming performance chamber. So I'm going to go through this. I don't know if any of you have done this sort of experiment before. Some not. Okay, good. So this is just zooming in on this picture. This is a homemade but standard swimming performance chamber. So we have a tube here. That's where the fish goes, in here. This orange and black thing here, this is the pump. It pumps water along this tube into this container through the straws to equalize the flow inside the tube. Okay? So then the fish is in here, and we can control the velocity or the speed of the water going through that tube. And we can increase the velocity. And that's just what we do to calculate critical swimming speed. You place the fish in the swimming tunnel, and you incrementally increase the velocity of the water. So every 10 minutes, we increase the speed of the water. So we're forcing the fish to swim against the current. And every 10 minutes, we make it go faster. And we're testing to see how long it takes for the fish to fatigue. At what velocity and how long of an interval did the fish last in this swimming tunnel. So when the fish gets tired, it goes and it rests against this mesh. We gently prod it, because some fish are really lazy. They're like, hey, I can just go lay against this mesh and not have to swim, but they actually do have the energy to keep going if you prod them. So we do gently prod them to see if they can keep swimming. If they can't, we end the experiment. This is a non-lethal experiment, so we reduce the velocity in the tunnel, and they just slowly recover from their fatigue. But what we get is critical swimming speed, and this is a proxy for understanding aerobic capacity, or how much energy do these fish have to do energetically expensive activities like swimming. These are the results. Again, I'm just showing you 2 of the species. Pugnose shiner, this is our endangered fish. We have the treatments on the x-axis and UCRIT, critical swimming speed, on the y-axis. There was a clear decrease in critical swimming speed in the Pugnose shiner that were held in turbid water compared to those held in clear water. Okay, so this suggests that their aerobic capacity is lower, that they just aren't able to sustain that energetically expensive swimming behavior, as well as those that were held in clear water. Black-chin shiner, mimic shiner, black-nose shiner, again, showed no difference between clear and turbid held fish. A couple things to point out about this experiment, and I meant to do this earlier on for all of the undergraduates in the room. All of this work was helped or even done, for the most part, by undergraduate students. So their contribution was really great to this experiment, and all of the 3 students that worked on this project are now going on and doing graduate school at some really good schools around the world. So that's kind of fun doing that. Another thing about this experiment, though, is that the fish were held under clear turbid conditions, but were tested in clear water. Okay, so that little fish is in turbid water for 2 or 3 months. It's alive, so it's a sublethal effect. But then we take it and we throw it into clear water and ask it to swim. We don't know what effect that has on the fish. So an additional experiment that needs to be done is to run the same test using turbid water in the swimming chamber as well as clear water. So take the same fish, run it under clear water, run it again in turbid water, and look at those differences. It'd be interesting to see if the fish held in clear water show any sort of immediate response to turbidity. One thing we noticed with the fish, but didn't really quantify at the time, was that if, in the course of the experiment, a fish in a turbid treatment died, their gills were completely clogged with a gray mucus. And the clay that we were using was a bentonite clay. It's this fine, fine gray clay. They seemed to have the ability to slough off that mucus. Again, we didn't quantify it, so it's another question about whether they were just building up mucus to protect their gills from turbidity. We don't know. But still, we see that the Pugnose shiner, the endangered species, shows this reduced swimming performance under low levels of turbidity. So, as I said, we started out comparing species at risk with non-threatened relatives, and found something out about how they respond to turbidity, although there are a lot more questions, I think, than answers at this point. And then we wanted to look across life history stages within a species. So for this work, we actually used a different species. So this is the spotted gar. Again, I was working with Nick Mandrake and Loren Chapman on this project. So we wanted to know if hatching success varied in spotted gar treated with turbidity and in clear water. Spotted gar in general, I think, are really cool fish. I talked to some interesting fellows on the ferry over today who informed me that all gar should be killed. Yeah, I know. They thought that they eat bait fish and game fish, so they should just be removed. I think they're really cool fish. In Canada, they're considered threatened. They're at the very northern edge of their range in Canada. In Ohio, I believe they are listed as endangered, so very imperiled, threatened. What's interesting about gar to me is that they have these different life history strategies, different life history stages. They have adults will move into really clear, highly vegetated backwaters, still backwaters, to spawn. They have adhesive demersal eggs. The larvae hatch from these eggs within a few days. They're free swimming, but they're actually usually attached. I'll show you a picture in a minute. Their mouth is actually like a sucker at that stage when they have the yolk sac still, and they're attached to vegetation. The young of the year are free swimming. Juveniles, 2 to 3 years in males and 3 to 4 years in females, and you've got their adults. What's interesting is that the post-larval stages have the ability to breathe air. So they have a bilobed gas chamber. They can go to the surface. They can gulp actual air and do some gas exchange within that gas bladder. They still have gills, which they can use, and some really neat work was done showing, I forget who did it, showing that the gar will actually allocate more time and energy to air breathing when there's really low oxygen conditions in the water, so hypoxic conditions. They will air breathe rather than try to rely on uptaking oxygen that's low through their gills. What's also interesting about this is that we might expect turbidity to affect the life stages differently. So adhesive demersal eggs and these attached larvae, they're exchanging oxygen through membranes or through the skin. If there is a lot of sediments in the water, that can adhere to the eggs, adhere to the larvae, and could potentially suffocate them. Basically, they can't uptake oxygen. Whereas the older stages, post-larval stages would be able, under those conditions, to go gulp air, be air breathers. And so that might not affect their respiration. But they're ambush predators. So they float like logs up at the surface. They see their prey, and they grab them really quickly. It's really neat to watch. There's some great YouTube videos if you want to see gar eating goldfish. Wonderful. But their vision could be affected under turbid conditions. So it's just an interesting difference in the way the different life history stages could be affected by turbid waters. So these are just some pictures to show you. These are some eggs with embryos. Here they're attached to this spawning mop in the aquaria. This is the attached larval form. So they've hatched, they've got their yolk sacs, but they're actually sucked onto the side of the aquarium in this case. And then this is a little juvenile. His name was Ed. So we asked, again, a fairly simple question. Does sedimentary turbidity affect hatching success in spotted gar? So this was our initial work just to start looking at the effects of turbidity on different life stages. We received from the University of Louisiana embryos, fertilized eggs. So 24 hours post-fertilization, we received a bunch of eggs from several females and several males. It was just one big batch. We placed 50 eggs in each of 10 tanks. 5 tanks were randomly assigned the turbid treatment and 5 the clear treatment. And again, really low turbidity. And this one wasn't on purpose. We actually had a lot of trouble in this experiment keeping the sediment suspended. So turbidity on average was about 6 NTU. Again, everyone's like, that's not turbid. It's more turbid than the clear treatments, which were less than 1. And it ranged between about 4 and 20 NTU. So there was a lot of variation in turbidity. Every day so we received the eggs, we partitioned them into the different tanks, and then every day we counted the number of surviving eggs and hatched larvae to get hatching success, which was the proportion of fish surviving. Okay, so we have time, days on the x-axis, mean hatching success on the y-axis. And it was a fairly, in blue are the clear tanks, turbid, brown, fairly clear difference between the clear and the turbid treatments. It was about a 24% reduction in hatching success in the eggs and larvae that were held under turbid conditions. So, again, really low turbidity. In Rondo Bay and Point Pelee, where there are reproducing populations of spotted gar, the average turbidity during the reproductive season, so May and June, is 12 NTU. So we weren't holding them at the turbidity levels where they're actually found reproducing. However, we did see this 24% reduction. What's interesting about this is that a model was created recently by Young and Coops in 2010 that suggested that any reduction in fertility of spotted gar greater than 14% would prevent the recovery of this species. So here I'm showing just in the egg and larval stages a 24% reduction at 6 NTU. So yes, we have breeding populations at 12 NTU, that level of turbidity, but how well are those populations doing? Well, they're declining, so we're not really sure what we need to do other than clear up the water for them to recover. Okay, so I've told you about a few projects. They were all sort of initial projects that we wanted to get going to try to understand what turbidity effects are on imperiled species in the Great Lakes. As I said, I also have some other research projects going on in Africa looking at the same stressor, looking at turbidity that is caused by humans, but in a group of fishes that are coping with it. What I want to do now is just take a couple of minutes to quickly, because I've only been in Ohio for about 9-10 months, just tell you kind of where my research program is going. For some of you undergraduates, I am looking for grad students, so it's a shameless plug here for graduate students. So I'm going to be continuing some of this work looking at turbidity tolerance and effects in Neutropus shiners. I'm also interested in the effects of human-induced water color and clarity changes on North American fish diversity in general. I really like sunfish. I love working with sunfish. The males are so colorful in many of the species, but not all. And we know that in some really turbid and really disturbed habitats, there's hybridization going on. That's a loss of diversity. So I'm interested in understanding the visual physiology mechanisms that are going on across the range where these fish are found. I'll be continuing my African work. I'm already starting to look at other stressors, combined stressors like turbidity, dissolved oxygen, temperature increases, and how those stressors work together to affect populations. And finally, thinking about both these local and global effects of environmental stressors on fish diversity, I think is important to sort of think on a broader scale sometimes. And I'm going to end with this little graphic, just to again emphasize what I'm thinking about when I think about the Great Lakes and turbidity in general. So here I've added a school of shiners, bait fish, forage fish perhaps we call them, but they're bait fish. We've got the human dimension. We've got people who want to catch the big fish. The big fish want to catch the little fish. We throw in that algal turbidity that is very dynamic. The mat changes in depth. Wind can stir it up. It's moving. It's fluid. What are the fish doing? What's happening to the schools? Are they finding their prey? Are they finding their mates? Is this guy finding his prey? And is this guy catching his fish? I recently saw on the internet a quote by a Lake Erie fisherman. The fish can't see the bait on the fishing line. And as a visual physiologist, someone who thinks about color and visual signals, this really stuck with me because I'd like to know what bait is he using and can the fish actually see it? So this is kind of where some of my research will be going in the future. And I'll just end there and say thank you to all the wonderful people who make this work happen.
Definitely have time for questions. So.
For the turbidity experiment, where we're testing like the schooling behavior, the visibility of the water, with the resources available, have you considered using like kiddie pools that are simulate the lake environment, like you can't see through on the sides, there's light coming down from the top?
Yeah, so one of the things that constrained that experiment was our logistical setup. So we had tanks in a fairly small chamber with fairly bright lights, very little space, and just because of the lighting and the color of the fish and the depth of the tanks, we couldn't increase the turbidity as far as we wanted to and still accurately see where the fish were in the tank. So something like a kiddie pool or one of those big cattle ponds, what do they call it? Cattle tanks? Okay, yeah, something like that would be better. And also a school of 5 to 10 fish. I don't know, maybe it'd be more realistic to have a school of 20, something like that. Could you throw in a predator? And I don't know if animal care will let me throw in a predator, but you could throw in a fake predator and see what happens. Yep. Yep. So it's an idea for sure. Yep.
Yeah, I've heard a lot of things on these recently about plastic microbeads, a lot of hair products. How do those and other genetically produced versus digest, how do those affect turbidity compared to more organic?
Yep. So there's the microbeads, and someone sent me something recently because they wanted to do a study on turbidity using something like actually getting something like that and throwing it in. They have they're all the same size. They all have the same roughness, and so it creates a uniform absorption and scattering of light. That's not necessarily what you're going to get out with, certainly with algae and certainly with sedimentary turbidity. The shape and the size of the particles, as small as they are, actually is going to influence the way that light is scattered. Now, Are you meaning that these microbeads are getting flushed into the water and causing a problem?
In a lot of places, I think,
Yeah.
areas they went, made it able to have those in their product there, so.
Okay, so, yep, it could cause a different set of problems. It could interact in interesting ways, especially in areas where there's already turbidity. That's really interesting.
I know you've taken mostly an experimental approach here, but I think you're in a really good spot for turbidity. And if you were going to go anywhere, I think go to western Lake Erie and some of the rivers that feed into it are good places to study turbidity. Have you or anyone else looked at more historical, species range maps and historical turbidity data and see, if you can find maybe cut points where some species stop being there, and then you could go in and do some of your experiments and see if they do better at this, and then we have management targets. We can then say our goal is to lower the turbidity in this system to this to bring back this.
It's a really interesting idea. I'm not necessarily familiar with what turbidity data is available. I've talked with people at the Ohio EPA, and they said they've got years of data. I don't know if they have turbidity that goes back. I think they're like all their fish survey data and the environmental data that goes with it, the 70s, I think. If it's there, it would be a really cool thing to do, and to set those sort of management parameters. With the Pugnose shiner and the experimental approach, I don't think the managers were really interested in hearing, yes, you have to reduce turbidity to 2 NTU. It wasn't considered viable, so maybe there's some other level. It's an interesting idea. Yeah.
Did you happen to look at the lateral line of the fish to see if that was kind of clogged like the nerve inside of it?
It's a great question. And no, I didn't.
Okay.
I really wish I had. There's certainly evidence that without vision, fish can still school with their lateral line intact. So there's been some experimental work where they cut the optic lobes and then allowed the fish to go, and they can use their lateral line to maintain schooling. If the lateral line was clogged, could that have affected it? It's very possible, and I didn't do it. I need a student.
Good time for one more.
So at least the Victorian cichlids and the sticklebacks too, there's some evidence that they're kind of not like fully evolving to deal with turbidity, but that there is some indication of some adaptation onto it. Yep. Is there any evidence that's happening with the shiners or into those projects?
I have no evidence of that. The evidence that so many shiner species are disappearing and populations are declining suggests that they are not coping, they're not adapting. But that's all I can say. I have no real evidence. I have some interesting evidence with the cichlids I'm working on of adaptations to turbid waters, not the shiners.
We could, we'll start it up at 8 o'clock, so you have like 5 or 6 minutes if you want to take a quick step out. All right, we're going to get started rolling again here. So before I introduce our guest lecture for this evening, just a reminder that next week, actually, there isn't a research lecture and a guest lecture. It's our RU culmination talk. So we've got 10 RU students. I think they're all alive still. All 10 of them are still alive and kicking. It's still early. That's right. They got 1 week left. So 10 RU students will be giving their talks, 15-minute conference-style presentation talks a week from today. Because we have so many talks, we actually start that event at 6 o'clock. So normally we're 7 to 9. This time we're going to be running from 6 to about 9 o'clock. So please come up and listen to those talks. We've been meeting with those RUs for the last 4 weeks and hearing about their progress, and they're doing some really spectacular, great stuff. So I hope everyone can make it in and listen to those talks. And then again, before I introduce Jeff Tyson, I just want to go around the room and make sure all the professors are still alive too, and make sure everything's going all right. Dr. Simon, you have any announcements for your?
We have a quiz tomorrow, but other than that, it's good.
Okay, Dr. King?
Lectures in the morning, take down our disturbance experiment in the afternoon. Hopefully it's still there after that yacht I've heard about that's made a big disturbance with Alligator Bar.
You had an experiment on Alligator Bar?
Yeah.
And something hit Alligator Bar?
Somebody said it was a huge yacht. And I don't know how you plan all of this too.
Dr. Beatty?
We are working on writing some papers about eutrophication in Lake Erie, and so those are coming due fairly quickly.
Okay, hopefully that's not a shock to you guys. You know you have a paper due, right? Dr. Marshall?
Dining hall will be open a little bit early tomorrow, so we can get everything on the boat for
7:15 for North Bass, huh? And the kitchen staff is okay with starting a little early, or are they disgruntled? You check.
Don't make that enough.
Just pop.
Pop-Tarts? A research coordinator, Dr. Justin Chapman?
Oh, yeah, I think Matt and Kevin did a really nice job diving for us, really big waves, Monday through Wednesday, to point sediment, or, chambers on the bottom of the measured nutrients. So, tomorrow we go get them.
Dr. Beatty, you have a question?
My students would want to know, do you feel the waves when you're at the bottom of the lake?
No, that depth.
See, now you have human evidence.
They have to be very large.
What depth were you diving?
30 to 40, all three sites.
Okay. I've been dive on the north side of the island before, which is 12 feet of water. If you're not paying attention, the jet goes by, you'll get smacked into some rocks. So you do feel it down at that depth.
Not really.
Yeah. Pat, any announcements?
We're still looking for a few brave people to stick around for a few weeks and help us run the island. So we will employ you if you pass our
rigorous obstacle course.
Rigorous obstacle course.
Yeah, have you ever seen the show Wipeout?
Yeah. So if you're interested, contact myself or Kevin.
So
By tomorrow.
how many weeks are you talking about?
At least 3 weeks.
Okay.
Yes.
And then I also want to introduce, we have another visitor up from Columbus. This is Dr. Kristen DeVena. She's a new hire at Ohio Sea Grant Columbus office, and she's joined us. Kristen, you've been on the job for now a week. Do you have any announcements?
I know, for a week.
Yeah.
Happy to be here.
Happy to be here. She comes to us from, there we go, master's and PhD from University of Toledo, and she's just finished up, before coming to us, about 3 years of a postdoc down at OSU with Dr. Stuart Ludson, so we're happy to have her on board. Good. With that, I'm going to transfer over to our guest lecturer. So we have the pleasure of having Jeff Tyson with us today. Jeff is the Lake Erie Program Manager for the Ohio DNR. You've been in that position now, 2 years? Prior to that, he was a fisheries biologist with the DNR office out of Sandusky for 10 years, then 10 years as the supervisor for that office, and now the position he holds now as the Lake Erie Program Manager oversees not only the Sandusky's fisheries unit on Lake Erie, but also the Fairport Harbor fisheries unit. And I don't want to steal some of the thunder that's coming out of the status report, but basically it's those two offices that are monitoring not only the game fish in Lake Erie, but also going to be managing the bait fish. And, so these are the individuals that are going to set the harvest quotas, not only for the commercial fishery on the U.S. side, but also the private anglers. So that's a heavy load to be lifting out of those two offices. And so Jeff comes to us from undergrad and graduates work out of University of Missouri. And so without any more delay, if we could welcome Jeff Tyson and his talk.
Well, thanks for having me. And I did have a connection when I was back at the University of Missouri. I'd never seen big water like the Great Lakes until I moved up here. But my graduate advisor worked on Lake Erie out of the Ohio State University as well, Rob Hayward. So he's the one that kind of kicked me out the door and up here. So today I'm going to talk a little bit about the status of Ohio's fisheries and Ohio's Lake Erie fisheries. and try to weave in kind of the management perspective, what we do as fisheries managers on Lake Erie, the kinds of things that we have to deal with, how we assimilate information, and I thought this might be kind of useful for folks in the audience. We've got a lot of bright young students here that are doing a lot of really good research and kind of getting that research underpinning, but understanding how that research can then be translated into management action, I think, is extremely important, particularly for folks coming out of school, so that they can start to look at some of these management agencies and understand what we have to do. So I'm going to kind of weave a story of Lake Erie fisheries, Lake Erie fisheries management, and some of the challenges that confront us, and some of the questions that we need to have answered, like Dr. Gray was talking about today, relative to some of these human-induced disturbances in the system. So this is an outline of my presentation. First, I'm going to talk about Lake Erie and its associated fisheries, and then I'm going to go over some of the management principles and regulations that we use to manage the fishery itself. Then I'm going to kind of drill down into the Lake Erie fisheries programs itself, talk about our assessment and our research, and how we weave that together into management action to sustainably manage the Lake Erie fisheries resources. And then lastly, I want to touch on some of the other factors that are affecting fisheries and fish communities that we as fisheries managers don't have any regulatory control over. There's no management lever for us, so we have to work with partners. And that's a really huge point that folks should take away from this presentation. First, you've probably seen all this stuff before. Reuters probably drilled it into your head, but Lake Erie is the southernmost, warmest, shallowest, and most biologically productive Great Lake. It's obviously the furthest south. It does warm up quite a bit right now. Surface temperatures are right around 75 degrees Fahrenheit, which we've seen upwards of 83, 84 degrees, at least in some of the assessment surveys that I've been on in the past. So it gets pretty warm. But obviously it's the shallowest and by far the most biologically productive, not only from the phytoplankton level, but all the way up to the top predator level as well. I've got a counterpart in ODNR Division of Wildlife that's our inland fisheries program administrator, and we go back and forth pretty much on a weekly basis. He generally says to me, well, you've just got one lake. I've got many. And I say, yeah, I've got one lake, but it's many habitats, many fisheries, and many fish communities. So we go back and forth. He doesn't like that. He's always tried to trump me with the fact that he's got to manage 30,000 lakes in Ohio or whatever. So, but anyhow. But in essence, Lake Erie is a highly diverse environment. This is the bathymetry of Lake Erie. It shows the depth, and you can see the gradient, red being shallow, dark blue being deep, and you can see the gradient from west to east. And that gradient is reflected in not only the bathymetry, but also in the temperature, biological productivity, so on and so forth. And that really does set up a huge diversity of fish habitats that establishes diverse fish communities across the system. And then one other thing that I think is extremely important and fits right in with what Dr. Gray was talking about today is that most of Lake Erie's major tributaries are found in Ohio. And these large South Shore tributaries exert huge structuring influences on water quality in Lake Erie and also in regions of Lake Erie, and also drive kind of fish community responses as well. We've got stock development that's based on these large tributaries. We've got essentially predator-prey dynamics that are unique to these tributary outwelling zones. So that really does create that diversity of habitat across the Lake Erie system. Again, Lake Erie is a fish factory. Over 100 fish species in Lake Erie. Includes warm, cool, and cold water species. West end's dominated by warm water species such as freshwater drum, the Maronis, the white perch and the white bass, channel catfish. Cool water species are also mixed into that western basin complex. The central basin is generally dominated by the cool water species, the perkids, the commodity species, as we call them, the walleye and the yellow perch. And then as you move down into the eastern basin, you get into the fish community that's dominated more by the cold water species, the whitefish, the lake trout, the burbot. All of these support sport and commercial fisheries throughout Lake Erie. And another thing that Dr. Reuter says probably numerous times is more fish are harvested from Lake Erie than the other Great Lakes combined. Lake Erie is not unique in interjurisdictional fisheries management, but it is unique in the number of jurisdictions that are involved in fisheries management. Obviously, these fish don't respect jurisdictional boundaries. They are a shared resource, and what our other partners do can potentially impact the performance of our fisheries, so we have to work together in a consensus-based fashion to manage these resources. We've got 5 jurisdictions around Lake Erie that have Fisheries Management Authority: Michigan DNR, Ohio DNR, Pennsylvania Fish and Boat Commission, New York DEC, and Ontario Ministry of Natural Resources. And we all work together under the auspices of the Great Lakes Fisheries Commission and the Lake Erie Committee. I'm Ohio's representative to the Lake Erie Committee. Annually, we set harvest quotas and so on and so forth. And I'll step through the process that we use for establishing harvest quotas, what information we use to establish harvest quotas, and then how we implement those harvest quotas at the state level. And I guess the last point is the fisheries focus is significantly different among these jurisdictions. Generally in Canada, it's primarily a commercial fishery, a commercial gillnet fishery. In the U.S. states, primarily recreational fishery, although we do have a fairly significant commercial fishery in Ohio as well. With that diversity of habitats, diversity of fish communities, and diversity of, we have a diversity of fisheries in Lake Erie. This slide kind of shows photographs of the fisheries that we do manage. Over here on the left side, we've got the 2 primary commercial fisheries. We've got a trap net fishery in Ohio, 18 commercial licenses, targeting rough fish and yellow perch. We also have a spring and fall seine fishery that targets primarily rough fish. And then we've got a whole host of recreational fisheries that we manage. We've got a private boat fishery in Lake Erie, well over 5 million angler hours a year, targeted at fishing on Lake Erie. We've got shoreline pier fisheries, fall and spring tributary fisheries, 750 charter boat, licensed charter boat captains in Lake Erie, the largest charter boat fleet in the Great Lakes, and then various and sundry other fisheries. And I think that might be John Hagman right there, standing next to the ice shack. So. This slide right here kind of shows the distribution of the catch among our various user groups in Ohio. In 2013, about 10.2 million pounds of fish were landed just in Ohio, and that distribution has generally been 60% of the biomass of fish that come out of the lake are attributed to the recreational fishery, where about 40% is attributed to the commercial fishery. About 20% of that recreational fishery landings is associated with the charter boat fishery, but those fisheries do look different, and that's one of the advantages that we have, because we have various stakeholder groups that are competing for the same resource that is limited in space and time. So this is kind of the distribution of the landings across the fisheries. The recreational fishery primarily targets walleye and yellow perch. They're upwards of 90% of the landings for our recreational fishery. We do have a smattering of harvest of white bass, of smallmouth bass, largemouth bass, and other species as well. The commercial fishery that primarily lands rough fish, and that would be freshwater drum, common carp, white perch, channel catfish. The value of those landings is fairly significant when you're talking about 5 million pounds of rough fish coming out of the lake. But the high value commodity species is the yellow perch. About 35% of the landings from the commercial fishery are yellow perch, and then about 13% are comprised of white bass. So fairly decent separation of targeted species that these 2 stakeholder groups utilize. There is some overlap with yellow perch, which creates some user conflict, and that's what I kind of want to talk about, because that's at the core of fisheries management, is dealing with that, dealing with those allocation issues, and making sure that we've met our mission to provide fisheries resources broadly distributed among Ohioans. So the motivations for each of our fisheries here in Ohio are significantly different. We've got the recreational fishery over on the left-hand side, and really the motivation for that fishery is recreation. People are going out, having a good time, catching fish. The secondary motivation would be food, and then we do have a profit component in that we've got a significant charter boat fishery that operates in the system, and then there's also profit associated with other sectors of the fishery, such as hotels and stuff like that. We have about 300,000 licensed anglers that utilize the Lake Erie resource on an annual basis, and like I said, about 750 charter guides in Ohio. That generates around $800 million in economic benefit on an annual basis, and that's $800 million associated with all the costs that are attributed to that fishing trip. The commercial fishery is motivated by something significantly different. First and foremost is profit. They're landing fish. Yields are important to them. They're landing fish and selling them. They do it for a livelihood, and then secondarily, it's a food. So we have 18 trap net licenses, like I said. We've had 18 trap net licenses in Ohio since, I think, 1978. It's been a limited entry fishery. We've got about 30 seine licenses that we issue annually. About 6 of those are active. And the commercial fishery itself generates right around $10 million in economic benefit. And it's probably slightly more than that because this is dockside value. So there is an expander that you can apply, and it probably would be anywhere between 10 and 20 million that's generated on an annual basis. So when we have these diverse fisheries, we have to come up with some management principles that guide how we do business to make sure that we equitably distribute those resources to the stakeholders that are out there. So I'm going to cover some of the ones that we use for managing the Lake Erie resource. First and foremost, we've got a fish supply of these 5 commodity species, and we spend a fair bit of our time assessing the status of these stocks so that we can provide a safe harvest level to meet human demand and use, but not compromise the sustainability and productivity of these species into the future. And we feed these various fisheries. So the management principles that we use is, first and foremost, we want to establish healthy fish populations in Lake Erie that are sustainable through time. We want to be able to distribute the benefits that Lake Erie provides to our diverse our stakeholder groups as well. All of our decisions are essentially science-based decisions. We can't make management decisions in a vacuum, in an information vacuum. So we have to take all of the information that we have, build that up into kind of management-relevant decision-making frameworks that we can then use to affect successful management of the resource. The next guiding principle is interagency accountability, and this goes straight back to a number of issues, but first and foremost is the quota management system. We're managing a shared resource, and we've made a commitment to our other interjurisdictional partners that we will sit at the table and work together to make sure that we sustainably manage these resources. If one jurisdiction decides to go rogue, it could have a very negative impact on the other jurisdictions out there. So we want to ensure that we are accountable at the interagency level, particularly with respect to the quotas that we establish, but there's a whole host of other accountability issues that we have to address as well. AIS is one. And then lastly, efficient use of the resource. We have bycatch issues with both recreational and commercial fisheries, and those are things that compromise the efficient use of this resource. So we try to minimize the amount of bycatch and inefficiency associated with management of this resource. The second kind of big point here in how we manage the fisheries out there is this Policy 2. And Policy 2 was established back in 1988, and it's called the Utilization of Lake Erie Fisheries Resources. And I'll show you how we implement this Policy 2 in our management framework. But Policy 2 was initiated back in the 1980s, and it was in response to quota management systems coming into place. And we didn't have any way to establish how we would allocate those among different stakeholder groups. Well, internally and with the participation from stakeholder groups, they established Policy 2, which says that it's essentially individual sport fishers get the first opportunity to take the harvestable portion of a quota. And then after that, commercial fishermen may be allocated the remainder. And that's driven primarily by the size of the recreational industry or the sport industry, the 300,000 anglers that are out there pursuing recreational harvest, the economic value associated with those kinds of activities, and then also given that there is some separation between what these fisheries target, we thought that they could still coexist even with lower portions of the quota species allocated to the commercial fishery. So this is actually a huge guiding document for us because it gives us a way to allocate, is what it amounts to. And this obviously was very controversial when it was developed. It was challenged up to the Supreme Court by the commercial industry, obviously, because they're taking second fiddle here. But the Supreme Court did agree that 300,000 anglers utilizing the Lake Erie resource probably should have that first crack at the resource, and then anything that's left over should be allocated to the commercial fishery. So we've got these two different stakeholder groups, and we have different fishery regulations for managing them. For the recreational fishery, we have daily bag limits, we have minimum size limits, gear restrictions, closed seasons and areas, and then protected species, and those are all tools that we use to ensure that harvest stays within the quota that Ohio has allocated. For the commercial fishery, we have a different set of fishery regulations in our toolbox that we use. First and foremost, we've got limited entry. We've had 18 licenses, commercial trap net licenses in Ohio since the late 1970s, which is a good thing in that those licenses have persisted for almost 30 years in their current form, so economically they're viable. And also, if you open that up, most open entry fisheries, you generally end up overcapitalizing, and none of the licenses can sustain into the future. So also we have some gear restrictions, closed seasons and areas, protected species, minimum size limits, and then the last thing here is individual transferable quotas. So that's how we implement the quota management system at the commercial fishery level. So each individual license receives an individual transferable quota that they can then harvest on an annual basis based on what that overall quota is. And I guess one point that I wanted to make before I got too far along the management process is really we only stock steelhead in Lake Erie. That's really the only species outside of the lake trout down in the eastern basin. We did stock some down in the west and central basin last year and the fall prior, but primarily steelhead are the only species that are stocked. The rest of the critters out there are sustained by natural reproduction. We don't control how many babies come out of the system. Mother Nature does. So that's a really important point. Okay, so next I'm going to talk a little bit about our fisheries program and what kinds of information we collect out on the lake, how we use that to implement fisheries management to meet our ultimate goals and using those guiding principles. A little background on our stations here. We've got two fisheries research units on Lake Erie, one of them based out of Sandusky. Primarily has assessment and research and management responsibilities over the west end of Lake Erie, so from Huron, Ohio to Toledo, north of the international line. We've got Chris Vandergood is the supervisor, one of the REUs here is working with him. And then we've got 4 biologists that are stationed out of the Sandusky station. We've also got a research vessel operator, OSU term position that was a former REU here as well. And then we have administrative staff over there. We have another station based out of Fairport Harbor, and that station has primary assessment, research, and management responsibility over the Central Basin. Kevin Kale's the supervisor there. We have 4 biologists, a research vessel operator that we're just hiring right now, another OSU research assistant, and then administrative staff. We have 2 primary sampling platforms that we utilize for a lot of our assessment and research surveys. in the west end is the RV Explorer, 53-foot aluminum hulled trawler that we use fairly extensively. And in the central basin, we have the RV Grandin, which is a 47-foot steel hulled trawler that we do a lot of our assessment and research work off of. So we're fairly well equipped. These are the largest vessels that we have. We do have a fleet of other vessels from 30 foot down to 18 foot that we utilize as well. Now I'm going to talk a little bit about our primary stock assessment programs. We spend a lot of time on assessing the status of stocks so that we can then develop management tools to efficiently manage those resources. And some of the primary ones that we administer out of both offices are a harvest assessment program, and that's our creel program. I don't know if anybody in here has ever participated in our creel program, but we've got a lot of creel clerks from years past that are running around out there. We've got, on an annual basis, we have 6 creel clerks that work Ohio's Lake Erie shoreline, 6 areas that they canvass, and it's an access point creel survey. They go from location to location, estimate effort, fishing effort, and then also estimate harvest by species and age. So ultimately, our harvest assessment program generates estimates of total harvest by species and age from Lake Erie on an annual basis. And this is just a picture of one of our creel clerks getting some information from one of the anglers. That's kind of the cornerstone of the interagency management. All the other management agencies around Lake Erie are doing exactly the same thing. They're estimating recreational harvest from the lake on a species and age specific basis. Same thing with the commercial fishery. Additionally, we have some population assessment surveys, and I'm going to talk just kind of briefly about 3 of those, actually 2 of them. The first one is our bottom trawl assessment survey, and a lot of you may have heard some things in the past about what's the hatch look like. Well, this is the survey that we use to estimate what the hatch of walleye and yellow perch, as well as a whole host of other species, looks like on an annual basis. Go out and sample 40 stations across the West Basin monthly from May through September, and then essentially generate a relative abundance estimate of the number of young fish in the bottom trawls, and then we use that to predict how many older fish will be coming into the population 2 years down the road. And this is a picture of our bottom trawl. It's a side scan sonar image, and it may not look all that cool to you, but I spent 10 years kicking this net off the back deck of the boat, never knowing what it looked like. And once we got some side scan sonar technology out there, this is the neatest image I think I've ever seen. Here's the doors right up here, which essentially spread that trawl out, just like the ones you guys kick off the back deck of the Gibraltar 3. Here's the head rope right here. Here's the cod end of the net. And there's some little fish up here swimming around that's getting herded into the net. You can see the tracks here where the doors are dragging along the sand bottom. So this is one of our primary assessment surveys for estimating the hatch. And it's not just the hatch for walleye and perch. We get relative abundance information for a whole host of other species out there. Second assessment survey that we spend a lot of time administering is a gillnet assessment survey. And we sample 50 sites, 50 some odd sites from Toledo all the way down to Conneaut with gillnets, and primarily targeted at assessing relative abundance of walleye and white bass. And we use that. Again, we've got other interjurisdictional partners that are administering a very similar survey as well, and we're all going to fold that in together. But here's kind of what our gillnet assessment survey looks like when we're pulling fish over the rail, and not a lot of fish in that net, but it is one of our primary assessment surveys. We've also got a couple of community assessments that we conduct on an annual basis. One of them is a forage assessment program. Separate from our bottom trawling, we conduct a lake-wide hydroacoustic assessment survey to estimate relative abundance of pelagically distributed forage fish in Lake Erie. And all of the jurisdictional partners participate in that exercise. We also have a nearshore community assessment, and here's a slide or a photograph that shows the electrofishing boat with the spheroids that we're using for shocking, but we go out and sample a number of stations across the near shore of Lake Erie to characterize the fish community, and then also collect a whole host of environmental water quality information as well, trying to look at explaining some of the trends and changes that we're seeing in that fish community fairly recently, actually. Lastly, all this information is summarized on an annual basis in our status report. We put that out generally in March of each year, summarizes all the data that we've collected from the year prior. And this is just kind of a picture that shows what our data, our long-term assessment data look like. We've been doing a lot of this stuff for a long time. The Creel survey has been administered since 1978, I believe, on an annual basis. And it's up here in the upper left-hand panel, just shows some of the data. On the y-axis here, we've got walleye harvesting millions of fish, and then on the secondary y-axis, we've got targeted angler hours. You can see that back in the late 1980s, we were harvesting, and this is just Ohio, upwards of almost 5 million walleye out of the lake. It's an enormous amount of biomass coming out. You can see that the effort tracks very well with it, so when we have lots of effort, we harvest lots of fish. That's what quota management systems are meant to address. So, and then you can see kind of a precipitous decline and a stabilization in not only harvest, but effort. So the harvest is largely driven by the amount of effort that's put out there in the system. And kind of to take that to the next level, what does Lake Erie look like relative, the fisheries performance look like in Lake Erie relative to historically back in the 1980s when the fishery was booming and everything was great? Well, we measure everything based on catch rates. So the amount of harvest doesn't really matter. Well, it does, but it's not a representative measure of fishery quality. You have to correct that for the effort that's out there. If you have zero amount of effort, then no harvest doesn't mean anything. So on an annual basis, we summarize the catch rates for walleye in Lake Erie, in Ohio waters of Lake Erie, our harvest rate. You can see in the 1980s, we had a phenomenal fishery. It was booming. We were harvesting 5 million fish. On average, we were catching about a half a fish per hour, and that's across all anglers across the entire year. 5 million fish coming out back here. Harvest rates kind of dropped a bit back in the mid-1990s. We had some spikes, and that was associated with strong year classes that came through the fishery, but generally stabilized at right around 3.5 or 0.35 fish per hour. And then we had a strong hatch in 2003, and you saw the catch rates in the fishery respond to that and jump back up over 0.6 fish per hour. But kind of what I wanted to point out is everybody talks about the booming 80s and how great things were back then when we were harvesting 5 million fish. Our harvest rates in the most recent years are as good, if not better, than what they were back in the mid-1980s. We're not landing near as many fish because we don't have near the effort. And then the second point is, let's put this into a regional context. What does Lake Erie look like relative to other upper Midwest walleye fisheries, Mille Lacs, some of the famous ones that are phenomenal? They have fishery management goals of 0.1 fish per hour. And an exceptional fishery is characterized as 0.15 fish per hour. We haven't seen that in Lake Erie. So this is an absolutely phenomenal world-class fishery in spite of these trends in harvest, effort, and even population abundance. Next, upper right-hand panel shows some results from our bottom trawl assessment data, and this is just walleye year class strength or hatches through time. And you can see that it's extremely highly variable. We had some very good hatches back here in the early mid-1980s, decent hatches in the 1990s, and then 2003 hatch, which was an absolutely phenomenal hatch, and it still persists in the system. In spite of the presumption that walleye live fast and die young, we see fish living out to 20, 25 years old. Our fishery right now is sustained by a lot of moderate year classes or small year classes here, but driven also by this big 2003 year class. 11-year-old fish are about 30% of the landings. And then the last one down here is our gillnet assessment, relative abundance time series. And you can see that abundance was higher back in the 1980s, dropped off, and as it stabilized, generally we're catching between 50 and 100 walleye per gillnet strap, which is about 1,300 foot in length. Now, I don't want to get too deep into the data here, but this is the kind of data that we use to help inform our management, particularly through the interagency fisheries management system. Ohio, Michigan, Pennsylvania, New York, and Ontario collect all this kind of information, and then we use that to generate fishery quotas or fishery catch quotas for both walleye and yellow perch. So all the information I presented above, as well as a whole bunch of other information, is used to assess the status of the stocks. We establish catch quotas, the quotas, to ensure that we have biologically safe levels of harvest. These quotas also do take into consideration social, economic, and political issues as well, but they don't vary from a scientific or a biological recommendation significantly. Interagency partnership, we've been doing this for over 30 years. 1976 was the first interagency quota management year for walleye. Yellow perch is a little younger. We implemented a quota management system for yellow perch back in 1996. And I want to show you kind of the process here. So we're taking a lot of this assessment information and we're building it into a management decision-making process. So we take all those surveys that everybody's conducting, we feed it into a statistical catch-at-age analysis model that generates a population abundance estimate, an age-structured population abundance estimate for both walleye and yellow perch. Yellow perch is, we have 4 management units, so we do it for each management unit. The yellow perch task group oversees that. They apply a targeted fishing mortality rate and generate a recommended allowable harvest. That information goes to the Lake Erie Committee, and we deliberate over the recommended allowable harvest, inform it by additional social, economic, and political insights, I guess, as well as other external ecological or biological information that we have, and we generate a total allowable catch, which is a safe harvest level. That safe harvest level is then divided up among each of the jurisdictions based on surface area in each of the management units. So this is an example of the West Basin. Ohio has about 50% of the surface area of the West Basin, so we get about 50% of the West Basin quota. And then, so this is all the science over here, right here. Sorry, a lot of stuff came in at once. We use kind of a risk assessment approach at the Lake Erie Committee level to evaluate some of these biological, social, and economic issues that will weigh into that decision. And then we move all that stuff over to our jurisdiction, to Ohio. So we take 50% of that quota, and how do we allocate that among user groups? And this kind of details that. Again, we apply Policy 2, which is the first screening level of internal allocation. So we estimate the sport harvest, and we use a series of multiple regression relationships to do that. We don't do a bad job of predicting how many fish are going to be harvested a year before they're harvested, but it's one way to estimate what that harvest is. Once we estimate what the recreational harvest is going to be in each of the management units, we allocate that quota to each of the management units for the recreational fishery. Anything that's left over is then allocated to the commercial fishery. Now, where we kind of potentially run into issues is if our quota for an individual management unit is lower than what we predict our harvest to be, we got a problem. So the way we've addressed that problem is we've actually built a bag limit framework or table into administrative code. So on an annual basis, if we get a quota allocation that is below what our fishery is going to harvest, then the bag limit is reduced based on some analyses that we did looking at our creel survey data to ensure that our recreational fishery does not exceed that quota. And then, so we go through all this for the recreational fishery. we allocate to the commercial fishery, and then it's pretty easy for them. We just split that out based on those individual transferable quotas. The percentage that each license gets is based on historical performance. So that's how we kind of divvy up the quota among all the various stakeholder groups in Ohio, particularly for yellow perch. Walleye, we don't have to worry about it. They are not a commercially available species in Ohio. Ontario allocates pretty much all of their walleye and yellow perch quota to their commercial fishery. Harvest quotas are used worldwide for managing commercial fisheries. Lake Erie is kind of unique because we have both commercial and recreational fisheries, and they're rarely used to manage sport fisheries. But they are used in Lake Erie, and it's kind of a unique way to implement it. Other jurisdictions have also incorporated this bag limit framework into their regulations as well to ensure that they don't exceed quota also, because everybody takes that interagency commitment seriously. And then I guess the last point is quotas are they're a cap, not a target. We don't want to exceed them, but if we don't get there, that's not hurting anybody. We can leave some fish in the lake, let them grow, harvest them next year. So that was kind of a thumbnail of the assessment side of things. This is a real quick thumbnail of research programs that we administer through the Lake Erie program. We've got a whole host of partners that we work with: University of Toledo, Ohio State University, Michigan State University's Quantitative Fishery Center, Great Lakes Fishery Commission, looking at a whole host of things: lower trophic level impacts, year efficiency, stock discrimination, understanding who contributes what to the population at what percentage, what drives recruitment mechanisms, can we predict how good the hatch is going to be based on a whole host of independent variables, weather impacts on short and long-term trajectories for species that we manage, and then adult movement. Some of the stuff that one of the REUs here was working on is looking at adult movement, particularly of walleye in the system. So those are kind of some of the themes that we work on, but here's a much broader kind of look at our research program. We kind of have three focuses. One is a mechanistic understanding. Why is something happening? So that's kind of almost a very basic information gap kind of research project. Some of them we have very specific questions like what is X to inform the model. One of the things that we're using the acoustic telemetry and tagging work to do is to estimate natural mortality rates for walleye, age specific or spatial natural mortality rates. We feed that value directly into the stock assessment model and then turn the crank. And then lastly, we sponsor research that develops predictive capabilities so that when X happens, we can expect Y. And all of this, when we go into research projects, we're constantly thinking about how we're going to take that and incorporate it into a management decision-making structure. So last, I want to just real briefly touch on other factors that affect fisheries and fish communities. I talked a lot about harvest. You know, we manage the heck out of harvest, but there are other things that kind of drive what happens out in the lake and what kinds of products we can provide to stakeholders. Habitat is a big one. Non-native species also have a significant impact. And then weather. And these things are kind of ordered based on where we have direct management levers. Harvest and stocking, we can do something about that. We can regulate the harvest. We can throw fish in the lake. That's where our management authority lies. Habitat, non-native species, we have some management authority there, but we have to work with external partners for the most part to address these kinds of issues. So we're kind of broadening the net that we have for managing the Lake Erie fisheries resource. And then weather, nobody can control weather or climate. So understanding that is really important. And this slide just kind of demonstrates that we spend an awful lot of time in developing these harvest strategies or stocking and stock enhancement. Don't spend a lot of time down here in the management of productive capacity, ecosystem management, or habitat because we as fisheries managers don't have that authority. And this is one of my favorite quotes from Bradford Brown, who was with NOAA, is one reason that we as fisheries managers spend a lot of effort controlling fishing or harvest is because that's what the law allows us to do. I mean, it's that simple. So we hear We have a lot of comments from stakeholders about why don't you do this, why don't you do that. Ultimately, we don't have the authority, but we do have a way to do that. Well, here's our fisheries management quota allocation or harvest management, just to demonstrate that we do have a process for that. But we do have a way to work with other folks. Environmental factors that affect fish and fish habitats in Lake Erie that are outside of our purview are things like dams, dredging, cormorants, exotics, land use, nutrients, power plants. These things are all really important to the fish communities out there, the productivity of those fish communities, and the distribution of them, and those ultimately affect our fisheries. Again, we don't have direct management authority over those, but they're outside of our control. So collaboration is huge. We have to work with folks in Ohio EPA, U.S. Army Corps of Engineers, a whole host of other folks, USGS for research, U.S. Fish and Wildlife Service, to try to kind of move the bar to meet our fisheries management objectives. And I'm going to show you a couple of examples, and this might make your head spin, but that's really what it does to mine too. Because and this is one that it's a Ballville Dam. It's a large dam on the Sandusky River. The dam has pretty much outlived its useful life. It's an impediment to upstream fish movement and has been for over 100 years. We've got a significant walleye spawning stock that utilizes the Sandusky River and spawning habitat just downstream here. That spawning habitat is degrading through time. As water comes over the dam, it's picking up the gravel cobble that was originally there, hauling it downstream, and so the spawning habitat's getting smaller and smaller, and we think the Sandusky River stock is responding by getting smaller and smaller. So there's a simple solution. Just pull that dam out, right? Well, the ecology and science really supports that, but there's all kinds of external complexities associated with that, and we don't own the dam. It's not ours to pull out, so we're going to have to work with partners. When you work with partners, you have to consider things like economics and cost. Ballville Dam, we're pretty fortunate in that we've garnered enough internal and external funds that we should be able to help the owner, the city of Fremont, pay for the removal without incurring any financial burden on the citizens of the city of Fremont. But that's only one hurdle. There's other things out there, alternative uses. How about turning this into a hydroelectric generating facility? It was originally built for hydroelectric generation. Why can't we use it now? What's going to happen with social or property values when you pull this dam out? We've got a significant number of folks that don't want to see the dam go away because they like that lake in their backyard. We've got permitting issues that we have to deal with, other ecological consequences. There's 800,000 cubic yards of sediment behind that dam. How do you pull it out and minimize the impacts of that sediment downstream? We've also got this degrading spawning habitat downstream. What will be the impacts if you pull it out in the wrong way and that slug of sediment embeds the slug of silt embeds the substrate downstream? Other ecological consequences. Lots of diked wetlands downstream that have concerns about this sediment slug coming downstream and affecting the marshes, which are diked. I don't quite understand that. Historical preservation, et cetera, et cetera, et cetera. This is a 100-year-old dam. You can't just pull it out and not recognize the historical consequences of it. So we have been working on this. We've been working with our partners with U.S. Fish and Wildlife Service, Army Corps of Engineers, City of Fremont, Ballville Township, Ohio EPA, historical preservation. Lots of complexity, right, for a very simple solution. But that's because we don't have direct management control over it. Another one is water quality. We really have no authority over this one. We ultimately manage at the end of the pipe. We deal with the water that comes in, and the fish community reflects that. And it does affect our ability to provide the kind of product that stakeholders want, like walleye and perch. And this graphic right here kind of just shows some of the trends that we're seeing. In a nutshell, and I've probably gone on for too long, but in a nutshell, what we're seeing is not a huge increase in one of our assessment surveys. This is a gillnet assessment survey. This is the catch of fish across time from 1994 to 2012. It's kind of variable through time. No significant trend, although we've seen significant trends in soluble reactive phosphorus, blue-green algae, and those kinds of things. So we're not getting more fish with more phosphorus. What we are getting is more eutrophic tolerant species. So we're seeing a shift in that fish community. We're growing lots of channel catfish, lots of common carp, lots of white bass and white perch, which are not what our stakeholders want. Ultimately, this water quality issue is driving what we can provide to the stakeholders. Lastly, other factors that are affecting fisheries, we've got a number of aquatic invasive species, and we're working, at least through our channels and with our partners, to work on the prevention strategy. Once things get in, all you can do is understand or try to understand what their impacts are. You can't predict them, though. And then lastly, weather impacts are absolutely profound. We have no control over those, and those affect hatches, they affect fish behavior, and they affect fisheries. So we go from lots of control to almost no control, and all of these things are extremely important from a fisheries management standpoint. So kind of in closing, Lake Erie fisheries are very important. They must be managed. Lots of complexity associated with it from the interagency standpoint, from the diversity of fisheries, fish species, fish communities, and stakeholders, but they have to be managed, and we have to come up with a management strategy that utilizes the science that we're generating on an annual basis. So in summary, Lake Erie supports these diverse habitats, fisheries, and fish species. Management is really complex. I gave you probably more than you wanted to hear about that. We have very limited direct tools. We can control harvest and we can control stocking, but lots of external factors affect our product. And then one other thing, it's not just about the science, it's not just about the research, it's about building that into policy, management, and people in action. And these partnerships are absolutely critical. So fisheries management, again, is not just out there squeezing fish, even though that's what I got into it for. I started out as a little wee wart, you know, whipping the James River with it was a Shakespeare, probably, riding my bicycle down on the river and fishing, but that's why I got into it, and it's still why I love it, is touching fish, but it's ultimately about people, stakeholders, and partners, translating science into management action and decision making. So research is extremely important, but you got to take that next step and build it into a way that managers, or a management structure that can be used to make decisions. And then lastly, collaboration across management authorities. So working with all our partners that do the things that we can't do. So with that, I guess I hope I have some time to take some questions. I've never worked in an exotic location.
We do it just so they time for questions.
You said that steelhead you only fish the Ohio areas in the lake. Do Pennsylvania, New York, or Ontario, do they
Stock steelhead as well? Yeah.
or other fish?
Pennsylvania does stock a few brown trout. That's primarily the only two species that would be stocked outside of lake trout restoration. And as far as the steelhead stocking, Ohio stocks about 400,000 steelhead on an annual basis. Pennsylvania by far stocks the most. They're dumping right around 1 million fish in a year. New York does stock a fair number. Ontario and Michigan obviously don't stock quite as many. So.
Do you account for in your preliminary quota basically illegal fishing?
Illegal fishing is theoretically either captured within our current assessment or our current creel survey. The way the access survey, the access point creel survey is designed, we can't discriminate between legal versus illegal harvest. We're counting boats as they go out and come back in, so we are capturing it to some degree, but that's not real satisfying. Part of another part of that is also captured within the variability in our natural mortality rate estimate. So now with that's with a survey, with the creel assessment survey. With the census, which our commercial fishery is assessed through a census, it's self-reporting. we don't capture that. And we've had some issues in the past with illegal harvest in the commercial fishery that have compromised our models.
So, I'm kind of a stupid question, but you mentioned that for assessing stock fish, one must use bottom trawl, right? Now, I'm more familiar with the marine aspect of this. I know that there's been a lot of talk about the effects that bottom trawling has on the vested community, about specifically on vertebrates and fish nesting plants. I was wondering if there was any knowledge studies done about the effects of bottom trawling done in Lake Erie, or if it has been.
The negative impacts of trawling in the system. You know, there's not a lot out there on it. I think probably the impacts, particularly in a freshwater system in Lake Erie in particular, are less than they are in a marine environment because you're dealing with different types of habitat. Most of the west end of Lake Erie is either a mud or a sand bottom. So likely there are some impacts. I'm not sure how big those are, but we don't have any idea. We haven't taken a look, and you can obviously see when you drag that bottom trawl in sand, you can see those tracks and you can see the tickle chain. So it is disturbing the bottom to some degree.
Especially because we just learned about turbidity and the effects on capture rate.
Yeah.
Why does Canada go all commercial and no civil sport fishing?
Well, it's how their fishery operates. They don't have a extremely large recreational fishery up in Canadian waters. Ultimately, it's each jurisdiction's decision about how they allocate that resource. So they can do whatever they want with it as long as they don't exceed the quota. But in reality, their recreational fishery is relatively small. They've got about 5 ports really that they operate out of. They're not like Ohio that has ports distributed from Toledo all the way. Fisheries operate out of another country.
I have a question more for students' benefit here. Skill sets that you're kind of looking for when you're hiring people to come into the program. What things do you like to see coming from the students? And are there any other kind of like jobs that you could recommend that they get, like internships or things that they should do to kind of prepare them for a career in business?
Sure. And I guess I'll start with opportunities. I talked about staffing at each of the offices, and those are all our full-time employees. But we've got lots of opportunities on an annual basis to participate in the annual creel survey, which is, we're hiring folks on an annual basis for that. Also, we hire on lab boat tech position at each of the offices that get to go out and do all these assessment surveys with us. And it's top to bottom. I mean, you're gill netting, you're trawling, you're conducting lower trophic level assessment, you're doing laboratory analysis and stuff like that. So that would be another opportunity. The OSU research assistant position is a 2-year term position. And the idea behind the creation of that position was to hire in somebody with an undergraduate degree that was considering going to graduate school. We wanted to bring them in for a 2-year term, train them in kind of the techniques and kind of how we think about information that comes in and build it into management strategies. Then, and build that skill set, send them on to graduate school, and then hopefully they'd end up back in the state of Ohio working for us up on Lake Erie. So those are 3, opportunities that I would encourage everybody here to take advantage of if you're interested in so inclined in moving into kind of the management realm. Skill sets that we're looking for are a lot of the skill sets you guys are working on right here. It's basic biology, basic fisheries techniques, fisheries management type things, applied stuff though. Understanding how to age fish. We age an awful lot of fish. And knowing how to do that efficiently, I guess, a lot of the techniques you can learn on the fly, but being exposed to them up front is very good. So, and then I guess kind of the intangible is trying to think about research projects that you're conducting right now, or as you conduct them down the road, and build that into a management action. So information is really good, but if you can't act on information, then it's just that, it's information. So folks that are thinking about how to utilize information to affect some sort of management action is really useful. And it's hard. I mean, I think when I came out of graduate school, I had a little bit of it. I worked for a management agency for a number of years, but it took me a number of years to develop that kind of mindset, I guess, that ultimately we have to use this information for decision making. So think about that as you move on in your careers, and if you want to get into, a management-oriented career, think about how to utilize those kinds of information to affect some sort of management change or action.
I would too, not only, the exciting, sexy courses, the biology and the fish species courses, but stats.
Absolutely.
That model of yours, that's not just simple numbers. I mean, you're running a lot of statistical analysis.
Yeah.
So it's important to get some stats under your belt.
Yeah, it is. The core of what we do, so we summarize a lot of long-term assessment data, but it's driven by, a strong background in statistical analysis, solving the kinds of problems that we have to solve, like estimating how many fish are going to be harvested the year prior to being harvested, is driven by, multiple regression type analyses. Statistical catch at age analysis modeling is a skill set that we're looking for in folks. Somebody to be able to turn that population model crank, identify issues in that, identify issues in the assessment data that are going in, and say, we got to fix something here. And then being able to estimate external parameters like natural mortality rate, understanding how to do that from a scientifically defensible standpoint. So.
Great. With that, thank both Jeff. Just a reminder that, again, that the RU talks next Thursday, and it starts at 6, not at 7 next week. Thanks, everybody.